Hello, I'm Mike Parrish, Agriculture Extension Agent in Dinwiddie County, and I'm for the soil because it's the foundation to everything in agriculture.
Welcome to another episode of For the Soil, a conversation. I'm your host, Jeff Ishii, and joining me is Mary Sketch Bryant with the Virginia Soil Health Coalition. Dr. Eric Benfelt with Virginia Cooperative Extension Community Viability remains on vacation. Mary, I'm not sure. I think he's somewhere on the West Coast, but we hope that everything is going safely and normal, and he'll be back with us soon. We have a returning guest today. Would you reintroduce our guest?
Thanks, Jeff. Yes, we have Dr. Jernigan with us from Virginia Tech. She's an assistant professor there. The focus in sustainable and organic food systems, really focused in soil biological processes and helping folks understand that better. Has a research, teaching, and service appointment. So really happy to have you back with us, Dr. Jernigan. And, to get us started, we talked some last episode about what you would suggest for, say, your backyard gardener to do. And I'd be interested, we have such a diversity of production in Virginia. You also have your larger, I say large in quotes because we're not out in the Midwest, but your larger grain cropping systems, grazing systems. How, if there's a, more conventional grain producer who's realizing, that they want to become more in tune with the soil processes, and seeing that need, where do they start on such a scale?
Yeah, well, thank you again for having me to continue this conversation and working at a variety of different scales. What I would recommend is going back to those two primary soil health indicators. So aggregate stability and organic matter as being really good indicators of the outputs of the biological community. And through Virginia Tech's soil testing lab, growers at any scale can send in samples and have a nutrient analysis done. I believe it's an add-on to do the organic matter, but I would recommend always getting that organic matter reading. Just if you're sending in a soil sample, it's worth the $4 that it costs to get that done. And in terms of aggregate stability, at any size, they're formalized tests, but if a grower's just, again, trying to see, is this practice that I'm implementing improving my soil structure and the overall functioning of it? One thing that you can do, which I love to do as a demonstration at field days or, for fun extension activities, is you can take a basic plastic pot that has holes in the bottom, fill it up with your soil from, let's say, field A, and then another one from field B. And then I have these bins with, just white bins so that I can see the amount of runoff or soil that's falling through the holes in the bottom. And you pour the same amount of water into both pots, and you can literally visibly watch as the soil is falling through down into your catch bins. And just that nice visual comparison. Okay, so this field soil that I collected has a ton of soil that fell through with the water flow. That means it's going to have less aggregate stability. It's going to be more at risk for erosion versus the soil where maybe you do have that better aggregate stability.
I know that you spend quite a bit of your time, Dr. Jernigan, in the classroom with students, probably in the field with students, but also you spend a lot of your time in research. And my understanding is that one of your focus areas of research is with something called microarthropods and how they impact soil health. For our listeners, what are microarthropods?
Yeah, so I'll give you a bit broader perspective from the field of soil ecology. So historically, how soil organisms have been extracted from the soil itself is based on their body size. So what we as soil ecologists think about first is their body width. Are they microflora, microfauna, like your bacteria, fungi, nematodes, those really small organisms that are not visible with the naked eye? Then we have our next size classification, which is mesofauna, which is any organism with a body width between 0.5 millimeters and 2 millimeters. So still really tiny, and most of their life stages, you're not going to see them with the naked eye. You need some sort of hand lens or tool. So those are the mesofauna, meso being in the middle, right? And then our macrofauna are anything larger than 2 millimeters in body width. So your earthworms, ants, termites, organisms that are easily visibly seen. But going back into those mesofauna, those are the organisms that I primarily work with. So as a part of my lab, I have a really big Berlese funnel set up. So for these organisms, which in terms of abundance are dominated by columbella, which are commonly called springtails, and mites, but there's also proturans, diplurans, other organisms that fall in that group, but in much lesser abundances. But we use a heat extraction method because their natural biological response to heat is, oh, let me burrow down into the soil and stay moist and cool. But instead, we have the heat source above the soil and they fall down into a funnel where they then get pickled so we can count them later, pretty much.
I want to
Um,
mention to our listeners that on our previous episode, which you can find that previous episode at the website forthesoil.org, and we're talking once again with Dr. Ashley Jernigan, assistant professor at Virginia Tech. You mentioned, Ashley, a very specific web address, a website called chaosofdelight.org. And I went there and I clicked on the gallery, and I am seeing some of the animals that you are describing: springtails, columbella. Those are magnificent little critters, but they're tiny, tiny little critters.
yeah, they're about the size of a grain of sand, and they also fall into that category of microarthropods. So once, as a soil ecologist, we've covered the basis of size because that's how we're able to enumerate or count the organisms in the soil. Then we move into taxonomy. So any organism that falls into the phylum Arthropoda that is also in that size category in the soil is considered a microarthropod, as you mentioned. And columbellans, mites, those are the primary organisms that we see in our agricultural soils in terms of abundance.
Okay. Mary, I know you're chomping at the bit with a question, but I got one follow-up question before we move on. How do we encourage microorganisms in our soil?
I love that question. What I always tell growers or anyone is you have everything you already need in your soil, in your ecosystem to grow plants, right? That weed's going to grow there whether you want it or not. But instead of focusing on any microbial additives or trying to get in specific organisms, you should always just be encouraging your native biological communities. So making sure that you're creating a hospitable habitat for these organisms. They're just like us, right? They want food and shelter and proper moisture. So by doing those practices that we've talked about before, reducing soil disturbance so that they can create their little homes and the aggregates that they want to live in, and by ensuring that we're feeding the soil to feed our plants, right? Focusing on organic matter additions and keeping those living roots that are going to provide exudates and nutrients for these organisms to feed on is really critical.
Dr. Jernigan, I feel like we're seeing more and more different, I say soil amendments coming up. You have your different inoculants. It's like this whole marketplace. And does it play a role? What should producers know about that? And because some of them are expensive too.
Yeah, that's one of the reasons why I always initially start out by telling people you have everything you need, and any element that could potentially benefit plants can also be brought in through just the natural ecosystems surrounding your farm fields where maybe they have been, you know, removed from that ecosystem because of past management practices. So I I typically much more encourage just making sure that you're putting on quality food for the organisms in the soil. And by adding in compost, green manures, you're also going to be bringing in new microorganisms, microarthropods, things that will add to the complexity and diversity of your soil food web, which will benefit that nutrient turnover.
Actually, we always talk about the four core principles of soil health usually during this podcast and also at our website, forthesoil.org. We talk about the things to do, but what about the things we should avoid? What are the don't do's?
Yeah, so obviously the biggest one is you don't want to tear your soil up and really do intensive tillage. Obviously, there's certain scenarios where as a grower you have to really be able to balance weed control and other aspects of crop production, but reducing the intensity of tillage is the number one. Don't go in there tearing it up unless you have to, and don't leave your soil uncovered. Honestly, even with bare fallows, I'd rather see some weeds coming in to a field for a couple of weeks. Obviously, you don't want to let them set seed because that'll cause future issues, but letting something living constantly be on there and not just letting it be a dead ecosystem is the main thing.
Are there any soil microorganisms? Is all soil life good, or is there any bad soil life?
Good question.
Yeah, so from a scientific perspective, I'm always like, oh, we don't assign moral value to our organisms, right?
Okay.
Because it's always context dependent. But I'll answer that question from the perspective of a grower, which I'm sure is what your listeners are more interested in. And the primary goal of a producer is to produce the best crop that they can, right? So in terms of soil organisms, there's a lot of diversity, including things that can negatively impact crop production. So, from a microbial perspective, we have our plant pathogens, which can be bacterial, fungal, viral, everything like that. Some of those live in the soil. A lot of the plant pathogenic fungi and organisms that live in the soil don't necessarily utilize that feeding strategy, though, right? They don't have a living plant. They might also be saprophytic or having that ability to feed on decaying plant matter and organic matter in the soil. So, kind of going back to feeding your soil environment and ecosystem, if given a choice, we can see that some plant pathogens will choose to function saprophytically, right? Feeding on that decaying organic matter over attacking the plants as a plant pathogen, for example, because of the resources it takes to invade that plant. They're going to choose the easiest way to stay alive. But there's a number of other organisms that can feed on roots like larvae and grubs and negative things that we have to consider. But I always like to keep in mind that everything in the ecosystem is playing a role, and finding that balance is what we're aiming for.
Well, just a couple of minutes remaining. And once again, we're visiting today with Dr. Ashley Jernigan, assistant professor at Virginia Tech, specifically in the Sustainable and Organic Vegetable Crops Production Lab. My final question, Dr. Jernigan, is about inspiration. You mentioned in the previous episode that you were inspired by the FFA organization, but looking back on your youth and your professional career so far, is there any one person or maybe a couple of people that you want to call out and say, hey, thank you, you inspired me to be doing what I'm doing today?
Yeah, I'd say, I guess my number one inspiration was my grandpa. He grew up farming, and in my childhood, he had taken on several of their career opportunities throughout his adult life, but he did have a bit of land that he farmed, and I loved going out there. He always had a strawberry patch for me, and I could go sit and eat strawberries, cucumbers, and be out in the soil. And his love for the land and agriculture was really instilled in me, and he's just, yeah, been a really great inspiration to my life in general. Um, Academically, I was very fortunate to have a really great advisor in my undergraduate studies, who's primarily a weed scientist, Dr. Matt Ryan up at Cornell, and he really helped my career trajectory and helped my perspective on balancing weed management and soil health and organic systems. And he also was the one that introduced me and connected me with my PhD advisor, Dr. Kyle Wickings, also up at Cornell at the research station in Geneva. And they were both so instrumental in the development of my perspective on agricultural sustainability and opened my eyes to how research really should be conducted to high quality standards and for the application of the farmers.
Well, it's been a real pleasure having a conversation with you. I want to thank you for everything you do for our community at large, obviously the agriculture community, the horticulture community. Thank you so much for everything you do, and thank you for joining us today.
Thank you for the opportunity.
And one last mention, that web address, chaosofdelight.org. I think you'll be as amazed as I am at some of the photographs of some of the microbiology that is taking place in your soil. Mary, thank you. We'll see you later.
Thank you both.
And we would like to remind everyone that you can find the 4 core principles of soil health at the website forthesoil.org. For the Soil, a conversation is made possible with funding support from the National Fish and Wildlife Foundation and the Agua Fund. Other partners include the USDA's Natural Resources Conservation Service, Virginia Cooperative Extension, Virginia State University, Virginia Department of Conservation and Recreation, and partners of the Virginia Soil Health Coalition. Views expressed on this podcast are those of each individual guest. To download a copy of this or any other episode, visit the website forthesoil.org. And if you should have a specific question about soil health, just contact your local extension office, your local USDA service center, or a Soil and Water Conservation District office. Music used during today's program was provided courtesy of the Flip Charts, all rights reserved. For the Soil, a conversation is produced by On the Farm Radio in collaboration with Virginia Tech. I'm your host, Jeff Ishii.